ora, good afternoon. My name is Jackie Bay and it's my pleasure to bring to you the LENScience Senior Biology Seminar Series this afternoon, Secrets in the Seaweed. I'm joined today by postdoctoral fellow from the Alan Wilson Centre, Crid Fraser, and Professor Hamish Spencer, Head of Zoology at the University of Otago and also a Principal Investigator at the Alan Wilson Centre and the National Research Centre for Growth and Development. It is our pleasure today to announce that this seminar is a collaboration between the Alan Wilson Centre and LENScience Connect. The Alan Wilson Centre is one of the 7 centres of research excellence in New Zealand and is involved in research in molecular ecology and evolutionary biology. Named, of course, after Alan Wilson, one of New Zealand's most prolific and well-known scientists. Now, we have had a fantastic 3 weeks on the wiki with your responses to questions and your discussion answers and the feedback that has gone to and fro between Michelle, scientists and yourselves around those discussion questions. So where are those people who are contributing to the wiki? Well, they're right throughout the country, as you can see here on the screen, and it's fantastic to see those schools that are just continuing to contribute. Remember, there is that award for the school that has the best contributions over the year, and there's several schools there that are looking like they're going for that award. Horowhenua College in particular, Otumotai College, but it's fantastic to see some new schools up there, Logan Park, Whangarei and other schools that are joining in to the wiki. So keep on the activity, and if you're not brave enough at the moment to actually write your ideas into the discussion, go and have a look and see what people are writing, because the best way of actually getting out of the seminar series a significant improvement in your ability to understand and write as a biologist is actually going to be to contribute to the wiki. But if you're not quite brave enough to do that yet, go in and have a look and see what people are writing about and how they're writing. Now, of course, we have the Best Wiki Discussion Award, as we do for each seminar. So for the last seminar, we're pleased to announce that again, it is Horowhenua College, well done, but a different student, Ali from Horowhenua College. Absolutely fantastic response and answer with an in-depth discussion about Challenge 2. So if you haven't been having a look at the wiki, go and look and see what Ali has written there. Of course, she's following in quite a strong tradition that Horowhenua are developing as being the leaders in the wiki, but let's not make them the only schools that are out that is out there and leading the way. So there's a challenge out there. Dilworth, you're sitting here in the live audience today, and I know we have Otumotai College that have made some good contributions in the past, Rangitoto College. So Unlimited is also starting to shine as a school with some very good contributions. So let's make sure that they are going around. But Ali, congratulations, a very well structured answer indeed. Now, today's seminar, Secrets in the Seaweed, is going to look at an issue of climate change. Now, climate change is something that is in the headlines a lot. And it's not actually what Crid set out to do. We're going to have a little look at the journey of Crid's PhD, which she finished just about 6 months ago. She didn't set out to look at climate change, but in fact, she managed to find something quite significant out about the level of sea ice and therefore the extent of the sea ice, apologies, and therefore the extent of climate change in the last glacial maximum, which was around about 20,000 years ago. And that's actually helping us understand a little more about that period and the natural climate change cycle at that time. But climate change is a contemporary biological issue in New Zealand, and some of you might have decided to select it as a contemporary biological issue for your 3.2 task. It is certainly one where there are differing viewpoints and opinions, as is required there. Let's have a look at what's happened in the news headlines in the last couple of weeks. Just yesterday, the New Zealand Herald, the 9th of June, climate row undermining confidence in science, headline. The Dominion Post a couple of weeks ago, basic climate change science sound despite IPCC errors. The Otago Daily Times, ETS change offers opportunities. And so it is something that is there, and we're reading about it, and it's something that is an issue for our society. Oxfam last year came out with this statement looking at the research into climate change from a variety of different places and different sources. They made a statement and said that by 2050, around about 75 million people from the Asia Pacific region, that is the region that we are a part of, could, notice it's could, be forced to leave their homes because of climate change. And of course, that's relating to changes in sea level. So it's an issue, and it's an issue that is actually quite complex, well, in fact, very complex and quite confusing. And just yesterday, Sir Peter Gluckman, the Prime Minister's science advisor, gave a speech at the University of Victoria. And he was talking about climate change, and one of the issues he brought out was the difficulty that the public have in actually understanding the issues of climate change, because it's not simple, it's not black and white, it's not straightforward. The public is confused about what we know and what we do not know about the science, and is unsure about whether governments are justified in making their decisions, hard decisions, despite the science not being certain. So what I hope you get out of this seminar today is a glimpse into the world of a PhD student and the journey that Crid took over a period of 3 years. It will relate obviously to evolutionary biology and will strongly support the development of your understanding of concepts of evolution and change that you are looking at in terms of your curricula. But I also hope that it will help you to explore something of the nature of science, because even if we don't become scientists, and not all of us are going to become scientists, all of us are going to become active citizens, contributing citizens within our communities. And one of the decisions that the community has to make, one of the issues that the community has to make decisions about currently and in the future, are issues of climate change. The science is incredibly complex. Most people, even most scientists, would not understand all the detail of the science. So how do we therefore decide whether to trust the opinions and views that our key climate change scientists are giving us about what they do know? And by looking at the work that Crid has done, we're going to take you through a little bit of a journey that shows you the process that a scientist has to go through in order to actually publish and make a statement about the work that they have done. And it's when they've gone through that journey that we can know that there is some certainty about what they are saying. Okay, so Crid's journey started after she had done her undergraduate studies. If you're going to do a PhD, you either do a Bachelor of Science with honours, which takes 4 years and then go straight into a PhD, or you do a Bachelor of Science and go on to a Master's, another 2 years, and then from there into a PhD. And the video that we can see playing now is maybe the sort of thing it's about to play that Crid might have imagined if you'd met her and she said, well, look, you know, I'm exploring this, starting this PhD, and I'm going to be down in the subantarctic islands. And there you are. I think that's what you might have imagined that she was doing in the subantarctic islands. She might have been down there with the fur seals and the penguins, and that unfortunate little bird down the bottom there, which Crid will tell you is actually eating the feces of the other animals. So that's its place in the ecological circles down there. Well, no, that's actually not what Crid was doing. So if you were imagining South Sea, sort of subantarctic islands, all these lovely penguins, no. If we look at this next video clip, we'll see what she was interested in. What she was particularly interested in was this. Now, those of you who live in the South Island will be quite familiar with this massive bull kelp. In the North Island, we do have some kelp around the coast. It just depends on what part of the coast you're in. It's an incredibly large seaweed. And what Crid was looking at in her PhD was the phylogeography of that particular population of the species, Divillia antarctica, which is one particular species of bull kelp. Now, a PhD is a journey, and it's a little bit like having a job. So Crid's PhD was supported by the Alan Wilson Centre, and she was based at the University of Otago. And when you do a PhD, you have supervisors, and your supervisors, a PhD is like a job. So your supervisor is like the boss at work, okay? And a lot of PhD students in New Zealand actually have a stipend, a scholarship, so that they are effectively working. And so Crid had two supervisors, both principal investigators at the Alan Wilson Centre, John Waters and Hamish Spencer. So when you do a PhD, you're doing an investigation, you're part of a team. Your 3.1 investigation, if you're doing it, and I hope that you've been given the opportunity to take up the challenge to do your own investigation this year, is a mini version, a very mini version of a PhD. If you can imagine having 3 years to select an area of interest to you that is relevant to a scientific research group and go in and do that research. So Crid will take us through that journey that had her looking at the phylogeography of the bull kelp. And what you'll look at is how she used biotechnological techniques, how she had to link to concepts of understanding of evolution that you're learning about at school at the moment, and how she developed her skills as a research scientist, which has meant that she's now gone on, completed the PhD, and is doing a postdoctoral fellowship with the same group. Now, the scholarship exam asks you to use your biological knowledge, your understanding of concepts, to actually think about a situation, analyse things, and write a comprehensive answer using that knowledge to explain what's happening in a situation that you might not have seen before. And before you started reading the paper about Crid's work, I'm sure that you didn't know a lot about bull kelp and about the phylogeography of bull kelp. However, what Crid has offered for us at the end of this seminar is that she has released for you some as yet unpublished data, because the experience that we're going to give you, the challenge that we're going to give you, is to actually use your knowledge of the biological concepts, the information that you'll gain from hearing Crid and Hamish talk today, and to actually analyse that data yourself. Now, you will not be able to look up anywhere on the internet and find Crid's analysis of the data. It will be published in time, but it's not published at the moment. So this is a great opportunity for you to actually see whether you can use your biological knowledge to analyse some of that data. So the concepts that you're going to be needing to think about are processes and patterns of evolution. You know what a gene pool is. You've been learning a lot about the impact of selection pressures on gene pools within a population. You understand gene flow. You understand that we can have speciation events and that we can have extinction events, and that all these can have an impact on biodiversity. Crid required an understanding of how to use biotechnologies, and I'm sure she will tell you she learnt an awful lot about how to use biotechnologies in this 3-year journey. These are the biotechnologies that you're learning about. They're the biotechnologies that are basic to a huge cross-section of research. You heard about Joe Perry talk about these same technologies in the cancer group. So I hope you can see that you can have your scientific training in one area and quite easily cross into another area of research and take those same skills with you. So extraction of DNA, the polymerase chain reaction to amplify specific DNA targets, DNA sequencing, the analysis of variation in the genes within the population, and then using all that information to actually look at phylogenetic analysis and reveal how related are these different populations. Okay, so in terms of how does this link to the assessment tasks that you'll be doing at the end of the year? Well, obviously we've got here the applications of biotechnological techniques. It is definitely one where, again, you're being exposed to things and looking at a different situation. Processes and patterns of evolution is very much something that you'll hear a lot about here. The concepts that you're understanding are the concepts that have been applied in this research. Can't do any of this without an understanding of DNA and gene expression. And of course, if you have decided, this could be quite an interesting contemporary biological issue to look at, something a little bit different. So I'm now going to hand over to Hamish, who is going to talk to you about the natural climate cycles that occur within the long periods of time here on the Earth. So, Hamish, thank you very much.
Thank you, Jackie. New Zealand is a very exciting place to do evolutionary biology. And one of the reasons it's such an exciting place is because we have a really dynamic geological history. And in the picture that you can see on the screen now, you can see that even just 20,000 years ago, New Zealand looked rather different than it does today. And the crucial thing is that 20,000 years ago, we were in the middle of an ice age. And that, in fact, was the last ice age and the last glacial maximum, at which point the sea level was considerably lower than it is today. One of the consequences of that is that what is today the North Island, the South Island and Stewart Island were all one landmass. So you can imagine for terrestrial organisms it meant that you could connect with other populations much more easily than you could today. But for marine organisms, getting around was much more difficult because there was all this land in the way. So one of the questions that you might say is, well, how do we know that 20,000 years ago the sea level was much lower than it was? How do we know that we were in an ice age? And we can tell from having a look at the graph that you can see now. Now this graph, I want to just walk you through it because it's reasonably complicated. On the y-axis there on the left, this part here, is an axis that measures the ratio of two oxygen isotopes. And along the bottom here, the x-axis is the age. And I just want to point out to you that now is here and a long time ago is over on the left. And a long time ago in this particular graph here is 800,000 years. So how do we use oxygen isotope ratios to tell us something about sea level? Well, the crucial thing is that there are two common isotopes of oxygen that are found in the natural environment, oxygen 16 and oxygen 18. Chemically, they behave the same, but physically, they're ever so slightly different. Oxygen 18 has two more neutrons in its nucleus. What we do when we measure the oxygen ratios is actually look at those ratios in some marine microorganism fossils, some things called foraminifera. And foraminifera make little skeletons in which these oxygen isotopes are preserved. So we can look at those oxygen isotopes and we know something about how those oxygen isotopes behave. Because oxygen 16 is ever so slightly lighter than oxygen 18, it evaporates more easily. And so it goes round and round the water cycle more quickly than does oxygen 18. And during an ice age, that really matters, because during an ice age, more oxygen 16 is going around the water cycle and falling as snow or other kinds of precipitation on the glaciers which are on the land. Right? So during an ice age, more oxygen 16 is occurring in glaciers on the land and not in the sea. So if there's more oxygen 16 in the glaciers, there's going to be more oxygen 18 in the seawater. And so the marine organisms, these foraminifera that I was telling you about, are going to take up that oxygen 18 and incorporate it into their skeletons more than they did oxygen 16. So that's what this axis over here is all about. So if we look back at this graph here, we can see that the current sea level up here, which is in this line across the graph here, that's the current sea level, and we can use those ratios to infer just how much of the water was on the land and how much was in the sea. And we can see that 20,000 years ago, the sea level was very much lower than it is today, and therefore all the North Island, the South Island and Stewart Island were connected. The other thing I would point out to you about this graph is that 20,000 years ago is just here on the graph, very, very close to now. And one of the things that concerns scientists greatly today about climate change is not so much the magnitude of the change, but just how quickly it's going to occur. You can see there that in 20,000 years, the sea level changed by about 100 metres. But we're talking about changes in sea level, not necessarily of 100 metres admittedly, but changes in sea level that are going to occur over decades and certainly within a couple of hundred years, not 20,000 years. So why does all this matter to biologists? And Jackie's going to talk a little bit about that.
Thank you, Hamish. So that gives you an understanding of those fluctuations that have been going on for many, many years and are slow, and a link in to the idea that those fluctuations are now happening and awfully faster than they have in the past. Okay, so let's think about speciation, because the concept of speciation is something that you have been exploring in Year 12 and we'll look at more in Year 13, whether you've started it now or you will do it later. So we've got a model population here of orange fish, red fish, seems to be coming up red on the screen there, that are in a marine environment. And what you can see along the bottom here is that we have the seabed, which has got a little bit of a hump in it. All right, a little bit like it does. The seabed is not flat. It's just like the land. The land is not flat, the seabed is not flat. Okay, so what happens when we have one of these climate fluctuations? So we have a climate fluctuation here. It happened instantly, but of course it's going to take a very, very long period of time, tens of thousands of years. And what is going to happen is that we're going to get, as a result of change in temperature, we're going to get a change in the sea level. And so the sea level goes down as it gets colder, the sea level comes up as it gets warmer. So okay, so we've got a change in sea level here, a little bit like we had, as Hamish described, where at one stage New Zealand actually didn't have a Cook Strait. And so we have a landmass sitting here, which is actually created a geographical barrier. All right, so we now have a geographical barrier, and we have our population of little red fish, which are separated. We've got some on the left of the landmass and some on the right, or the east and the west of the landmass. Now, as you know, as you've just heard, this is going to take tens of thousands of years. Now, you're all studying biology so that you know that in a population over tens of thousands of years, there are going to be mutation events. There are going to be variations that are caused. So maybe those mutation events start to happen, and if we can see what's happened to the population which is on the screen there, we now have an orange population sitting to the left of the screen and a yellow population sitting to the right of the screen, representing that some of those mutation events and selection pressures over many, many tens of thousands of years may have caused quite significant changes in the two populations, to the point where when, and again, it happened instantly on our little animation, but of course it's not going to be instant, over a very, very long period of time, we get the continuous climate cycles, and we get the change in the sea level, and we're back to one habitat. And in this one habitat, we have a mixture of the two populations. If we find that those two populations are no longer able to successfully breed to produce fertile offspring, as you know, we have a speciation event that has occurred. So thinking about the basics that you know about allopatric speciation and about how climate change can be one of the factors that can influence a speciation event, that little model tells us a little bit about what's happening there. Now, Crid is going to talk to you about some of the other events that happen during changes in climate and climate cycles. Thanks, Crid.
Okay, so Jackie's talked about the effects of sea level change and the impacts that can have on animals. But of course, there's other impacts of climate change. We know that in ice ages, the Earth is a lot colder. There's a lot more ice around. Right now, most of the cold on the ice, is centered at the poles. Well, it always is with the Earth. And many plants and animal populations have very narrow ranges of environmental conditions that they can survive in. So you can imagine as the Earth goes into an ice age and the glaciers expand from the poles, many populations of plants and animals are pushed towards the equator or forced to move. And then as the Earth moves back into a nice warm interglacial period, those populations, those species can move back up into the area freed by ice, or the areas that are more suited to their habitat requirements. So that leads to a lack of diversity in the populations that are recolonizing the glaciated area. And that can often be seen today. This is an example of a study on the brown bear in Europe. It was done by some French scientists, Pierre Taboulet and Jean Bouvet. And they studied populations all around Europe. Those little spots, different colored spots, represent different populations of brown bear, and the different colours represent different genetic variations in those populations, different haplotypes in the mitochondrial DNA. So they related the patterns that they found to the presence of the ice sheet at the last glacial maximum, which covered much of Scandinavia and Britain. They suggested that as the ice receded, some bears were able to move up into those areas and rapidly multiply to fill that new area. So there's a lack of diversity in the recolonized region. And the way they could tell where those bear populations probably originated from is by looking at the phylogeny of the haplotypes that they found. So this is a phylogenetic tree, and you can see that all of these blue haplotypes here and here are very closely related, so that they know that these populations share some common ancestor. And similarly, the red ones here and here are very closely related. So it looks as though these ones have come from here. And Hamish is now going to talk you through the idea of phylogenetic trees a little more.
So Crid talked to you about a phylogenetic tree for the bear populations that she was interested in. So what is a phylogenetic tree? Well, a phylogenetic tree is simply a way of showing relationships among organisms. They may be individuals, they may be populations, as in the bear example that Crid talked about, or they may be simply different species. And we use, as evolutionary biologists, those phylogenetic trees to infer the evolutionary history of the groups of organisms that we're interested in. So let's have a look at an example. Let's suppose that we were interested in monkeys, bats, eagles and kiwis. Now you know how those four organisms are related to each other, those four species. You know that the monkey and the bat are both mammals and therefore each other's closest relatives, and similarly the eagle and the kiwi, both being birds, are also each other's most closely related to each other. But let's have a look at how an evolutionary biologist might try to construct a tree showing their relationships. One of the things that evolutionary biologists do are use things called shared derived characters. So what is a shared derived character? Well, let's suppose we were looking at that for a group of organisms, or the group of four organisms, and we might put the eagle and the bat together because both the eagle and the bat have wings, right? Now, the presence of wings is a shared derived character. It's shared, right? Both eagles and bats have wings, and it's derived, which means that those organisms possess it, but the other organisms don't. So if you were constructing a phylogenetic tree based just on that single character, that's the kind of tree that you might come up with. Now you know that that tree is wrong, right? That tree is wrong because you know that the bats and the monkey should go together and the kiwi and the eagle should go together. You know that that in fact is the right tree. As I said, the two birds go together and the two mammals go together. And the reason that evolutionary biologists would come up with that second tree is by looking at a whole suite of shared derived characters. So for example, the birds go together because they both have feathers. They both lay eggs. Those are shared derived characters. The mammals go together. They both have hair. They lay, they have live young, and they suckle their young, right? They produce milk. So those three characters are shared derived characters, and they unite those two species of mammal. So the other thing about phylogenetic trees that you might have noticed by looking at your textbook is that they seem to be drawn in all sorts of different ways. There's lots of different ways that a phylogenetic tree may be drawn. So one of the most common ways is what's called a cladogram, and a cladogram shows you those relationships. So here is a slanted cladogram, and here is a cladogram that shows you the same information but is rectangular. Now those two diagrams are just different ways of drawing the same information, right? But you might see both of them in your textbook. You might want to show slightly more information, and this is what's called a phylogram. And a phylogram is ever so slightly different because it has different branch lengths on it, and those branch lengths show you the estimated time that's passed. So a cladogram shows you the relationships, and that's about it. But the phylogram will show you the relationships, but also something about evolutionary time. And you can see in this tree, for example, that the estimate of how long the ancestor of D and E lived is a longer time than the ancestor of A, B, and C, right? Because the branch length here is longer than the branch length here, right? The other thing that you might notice is that time seems to move in different ways across the page. The standard way, or a standard way that's very common, is for time to move from left to right. So old is on the left and new, recent time is on the right. But you could turn that around and have it going up the page like an ordinary tree, so that time starts at the bottom and now is at the top of the page. You could even have it round the wrong way if you like and have it like a root. I've seen trees published like this. These are all the same tree. You notice that I've just rotated around, and the current taxa that are alive today are at the bottom of the page. And the final way that you might see a tree drawn is in a circle, right? And you often do this if you've got lots and lots and lots of species that you're trying to fit onto one page. So time starts in the middle and it moves to the outside. So I've talked about time, but how do biologists know about how much time has passed on an evolutionary tree? Well, basically they use versions of what's known as the molecular clock. And the molecular clock measures the rate of change at a molecular level. And there's different kinds of molecules that different biologists might use. So today, most of the time, we look at the changes, the number of mutations that have occurred in DNA. So both nuclear DNA and mitochondrial DNA. And mitochondrial DNA is mostly what Crid used, as you will see in a moment. But you could also look at changes in RNA. And interestingly enough, the original molecular clock looked at the number of changes in proteins. And Alan Wilson, for whom the Alan Wilson Centre is named, was a New Zealand scientist who was instrumental in applying the molecular clock as derived using proteins to human evolution. So that's where Alan Wilson Centre made his original mark. Alan Wilson made his original mark. So how do you work out how much molecular change has occurred? Well, first of all, we measure the number of changes in the DNA sequence or the protein sequence or the RNA sequence over time. And quite reasonably, if you're looking at two species and there are lots of differences between them at the molecular level, you think that they have been separated from each other for a long time, right? So the larger the difference between species, the greater the time since they split, the greater the time since they last shared a common ancestor. Okay, so that tells you about relative time, right? You know which species are more distantly related, have been separated for longer periods than others. But how do you convert those relative times to absolute times? How do we know that certain things took 5 million years or 10 million years? Well, what scientists do is that they calibrate their molecular clock. And there are different ways to calibrate the molecular clock. Mostly we use geological or fossil evidence. So we might have a fossil that we know fits somewhere on our tree, and so we know that that particular branch of the tree occurred, say, 5 million years ago or 10 million years ago. And we use that to calibrate how fast, if you like, how fast the ticking occurs on the clock, how fast the mutations occur. So another little bit of terminology here about our phylogenetic trees. We often want to talk about parts of the tree, some subset of the tree, things or groups that are closely related. And the word that we use is clade. So here we can see that species A, B and C are each other's closest relatives, right? They're together on the tree, and back down here at this point they share a common ancestor. And that group of species, that part of the tree that has all of them in, is called a clade. So there's one clade. There's another clade down here. D and E also form a clade. So you might want to be comparing those two parts of the tree. And you can also see that there's another clade here. A and B form a clade. They're each other's closest relatives. And the last thing that you will see on some of Crid's trees that she will show you in a few moments is you may see some numbers above or below the branches. And those numbers are a measure of how much statistical support there is for that particular branch, right? One of the things you want to know is how certain are we that, for example, that A and B are each other's closest relatives. And if we look at the numbers on this tree here, we can tell that. So the number above the branch here is what's called a posterior probability. And the simple way to explain that is to think the numbers range between 0 and 1, and if the number is close to 1, we're pretty sure that that branch really did occur in that tree. The numbers underneath that range between 0 and 100 are called bootstrap values, and the larger the number, again, the better. So you would like it to be over 90 if you could. So this value here of 89, which I've just made up, is not too bad, but there's a little bit of uncertainty that really A and B are each other's closest relatives. So basically you want these numbers that tell you about the statistical support to be big. So the last thing that I want to talk about is how we actually get these phylogenies from the genetic information that we have, right? So Quid talked about things called haplotypes. So what is a haplotype? A haplotype is simply a DNA sequence that differs from what's called a homologous sequence, which is just the corresponding piece of sequence in another individual or another species or another genus at one or more different sites. So it's basically looking at the same piece of DNA sequence in a bunch of different individuals. So here's an example here where we go and sample one individual and we, for the particular bit of sequence we're looking at, we get these 7 bases. And let's be imaginative and call that haplotype A. We go out and we pick another individual from our population and we get the DNA sequence for that, and it turns out to be the same, right? So those 2 individuals share a haplotype. We go and look at a third and we discover that at the second position, instead of that individual having a big A, they have a big G, right? So there's a difference there, and that's a slightly different haplotype. When we look at a fourth individual, it turns out that this individual has a number of changes in the corresponding piece of DNA in that individual's genome. So here's the kind of information that you have when you collect DNA sequences from a bunch of different samples. So how do you convert that to a tree? Well, the answer is there's lots of ways to convert it to a tree. There's about 5 or 6 different algorithms that scientists use. Ideally, the different algorithms would give you the same tree, right? You've got some information, you're trying to estimate who's related to what. It shouldn't matter exactly how you do that, and most of the time if you've got good information, it turns out it doesn't matter a great deal. But how would you do it, at least at a simple level? Well, clearly individuals that have the same haplotype are likely to be closely related, right? You know that you are genetically more similar to your close relatives than you are to people more distantly related, right? So you would put them together on a tree. The haplotype B, which is 1 base pair different, surely is going to be fairly close to haplotype A, and the last one that you think would come in the tree is going to be the one that's most different from all the rest. So that's roughly how you construct a tree, but as I said, there are lots of different algorithms that people use. So Quid is now going to talk to you about the organisms that she worked on.
All right, this is the bit I know you've all been waiting for, the bit where we talk about seaweed, which I'm sure you all love as much as I do. Yeah? All right, I worked on southern bull kelp. It's a genus of kelp that's very common around New Zealand, so you're probably very familiar with it. There are actually 5 named species in Davilia. 4 of them can't float, so they're non-buoyant species. Only one of them has the ability to float, and that's Davilia antarctica. And when we look at the distributions of these species, you can see that all the floating ones have very restricted distributions. Davilia podatorum occurs only around southeastern Australia, Tasmania and southeastern Australia. Davilia wallana is only around southeastern New Zealand. Davilia chathamensis is only found on the Chatham Islands, and there's another species that has yet to be described that's found only on the Antipodes Islands in the New Zealand subantarctic. Interestingly, the species that can float, Davilia antarctica, is found pretty much all around the southern hemisphere, at least down south in the southern hemisphere. So it's found all along the coasts of New Zealand, along the coast of Chile, and around all of the subantarctic islands. As Jackie said, you're probably familiar with it. You've probably seen it around the coasts of New Zealand. This kelp fan is a girl who worked with me for a few weeks over summer collecting kelp, and at the beginning she hated kelp. She thought it was slimy, smelly, horrible stuff. But by the end, as you can see, she was rolling in it. It's a very charismatic species, and you might even recognize it from the movie King Kong. There's a section where King Kong is chasing them and they're leaving the island, and King Kong's wading through the kelp and they fall into the ocean and they sort of swim around in the kelp. Anyway, maybe you have to be a kelp fan to recognize that. So Davilia antarctica can float because of the structure of its fronds, which are pumped up with gas. This picture on the left shows a cross-section through the kelp, and you can see that there's sort of honeycomb structure inside, and each of those vesicles is filled with gas, which makes it really buoyant. The kelp weighs a lot, so for this kind of kelp to float around the ocean is pretty amazing. It's also really tough. It can survive a lot of bashing against rocks and can survive a lot of thrashing around if it drifts out in the ocean. The other pictures show some of the holdfast to the kelp. The holdfast is the bottom part of the kelp that helps it to attach to the rock, and it's often filled with little burrowing creatures, worms and crabs and snails. So there's a potential for those animals to travel with the kelp if it breaks off and drifts around in the ocean. In fact, somebody sat on a boat once when they were traveling from Australia to the subantarctic, and they counted pieces of kelp going past. And from that study, they estimate that there's 70 million pieces of De Villiers Antarctica adrift in the Southern Ocean at any time. That's a whole lot of kelp out there. And I think we're going to show a little video of kelp drifting in the ocean. Yep. So as you can see now, there's some kelp rafts. This is us on the last subantarctic trip, and we tried to pick up some kelp at sea. It's kind of hard to chase down a chunk of kelp in the big waves in the Southern Ocean. You can see that's actually a big mass of multiple kelp individuals and multiple species of seaweed. So it was really exciting to be able to get hold of some of that. So what would happen if kelp broke off, for example, in New Zealand and drifted around in the ocean? There's a very strong current that circles Antarctica. It's called the Antarctic Circumpolar Current, and it goes around from west to east. It's the strongest current in the world, and it's pretty much unbroken all the way around. So you can imagine if kelp breaks off in New Zealand, it could easily be swept in this current around to somewhere like Chile or some of the subantarctic islands. So in my PhD, I set out to look at how well connected these populations of De Villiers Antarctica are by looking at the DNA of the kelp. Another way to express that would be, does De Villiers Antarctica disperse via rafting? Obviously, with kelp occurring all around the Southern Ocean, I had to get to quite a lot of sampling locations, and we didn't really have any money. Obviously, getting to the New Zealand and the New Zealand subantarctic wasn't too hard. You could just jump in a car and do the coast of New Zealand. And Heritage Expeditions, a tourist company, took me down to the New Zealand subantarctic, which is fantastic. Then I asked a British organisation to give me some money to go to the Falkland Islands, which they very kindly did, and I had to fly via Chile. So I hired a car there and asked a Chilean to come along and help me, and we collected kelp for a few weeks along the coast of Chile. South African National Antarctic Program also gave me trips to Marion Island and Gough Island, which are some of the most isolated subantarctic islands in the world. That photo in the corner shows me collecting on Marion Island, and king penguins sort of marching up behind. They're very curious. They don't see a lot of people, so they were coming in to see what I was up to. And I didn't get to the other islands, but I was sent samples from them. So in the end, I managed to get samples from Macquarie, Crozet and Kerguelen, South Georgia as well. So we covered pretty much the whole range of De Villiers Antarctica. So we've gone from the field, which is wet and cold, and I think there's going to be a video of me snorkeling in the subantarctic, which is lots of fun, and I recommend getting a thicker wetsuit than a 3 mil. And having a boat standing by spotting for sharks is also a good thing, or killer whales. There were often killer whales going past. So jump in the ocean as quickly as possible and get the samples and get out. And so we've gone from the field back to the lab, and things are a little different there. We try and keep the samples well organized and clean. This is what they look like when I've finished with them. It's got a label showing where the kelp is from, the date that it was collected, and what species I think it is. And I keep those in folders, as you can see, nice and organized, nice and easy to find which sample I'm after at the time. And they're kept dry to preserve the DNA. Then I start the process of DNA extraction. So I break off a little bit of kelp. I normally take about 1 millimeter and put it in a small tube with some Keelex solution. I incubated it at about 60 degrees overnight, and that separates the DNA from a lot of the other cellular gunk you've got. Spin it down and the DNA comes to the top, so we're able to take that off and start the process of PCR. And PCR, it's all very well to say you're going to do PCR and amplify some genes, but you have to choose which genes to target, which isn't always straightforward. As you know, you use primers to target the piece of DNA that you're after, but not all primers work on all taxa, which is sometimes a good thing. But you might have a set of primers that you want to work with the taxon you're looking at, and it might not happen, so they might not work. You might need to design new primers. And sometimes even once you've got primers working and the DNA amplifying nicely, the bits of DNA that you get back aren't very informative. They might be identical among all the individuals you're looking at. So you want bits of DNA that are also informative. So the way I chose my genes was a little bit hit and miss. I spent a few months in the laboratory mucking around, trying different primers, designing primers, amplifying DNA, and seeing what I got back. But finally, I settled on a mitochondrial gene, CO1, and a chloroplast gene, RBCL. So those were the two main ones I looked at. I also studied a nuclear marker, 18S. But for the rest of the talk, I'm just going to show you the results from the CO1. It was the most informative. We just used the results from the other markers to back up our results. Right, so you get back your sequenced DNA. It looks a lot like this when it comes back. It's a series of coloured peaks that show which bases are at each position. And you need to line those up for all your individuals. And this is kind of a snapshot of what it can look like once you've got all your data. This is only a tiny corner of the data. In fact, it would pretty much fill this whole room if you could see the whole thing. Down the left-hand side, you can see the code that identifies the individual that you're looking at. Generally, it's a code I made up that tells me which individual it's from. It has the site and the number of and the species and which number replicate it is. So this is a close-up of some of the data with the different bases shown in different colors. And you can see that at many bases, all of the individuals are the same. But for example, at this 5th base here, some individuals are Gs and some are A. So that's an informative site. And hopefully you get lots of informative sites that can show you genetic diversity among all the individuals. So once we've put this through the phylogenetic software, we get a tree back, or several trees, but this would be one. And you can see that this is just a tree showing Davilia antarctica, none of the other species of Davilia. And you can see there's quite a lot of genetic diversity just in this one species. And some of these are the numbers that Hamish talked about. So we can see that many of these clades are quite well supported. The clade for Chile, this one, has a very high support. So we're fairly confident that all those haplotypes from Chile belong together. New Zealand North, a little less well supported, but still a very high posterior probability value up there. So most of the clades were well supported. And we ended up with 4 main clades. One from New Zealand North, it's a region New Zealand North, it doesn't mean North Island New Zealand, actually came down as far as about Banks Peninsula. One from New Zealand South, one from the subantarctic. This one we think is a different species, so I'm not going to talk about that. And one from Chile. What got really interesting was when I plotted these haplotypes on a map, and immediately you can see that there's one haplotype that's very dominant, the one I've coloured in red. It's not only dominant, but it's the only haplotype found at most of the subantarctic islands. And that contrasts sharply to the diversity you see further north, so around the coast of New Zealand and along the coast of Chile, there's quite high genetic diversity. So if we take a closer look at the New Zealand region, you can see that along the South Island, for example, a couple of sites that are separated by only a few tens of kilometres are showing genetic diversity, not only among sites, but also within sites. This site has 2 haplotypes, this site has 2 haplotypes. And down in the subantarctic, the New Zealand subantarctic, you also see quite high genetic diversity with multiple haplotypes within and among sites. So what could explain the pattern that we're seeing where there's complete lack of genetic diversity in the subantarctic? Well, it looks very much like a recolonization event or a colonization event where that haplotype, those populations of kelp are only recently established in the subantarctic and have probably come from a single source population quite recently. We know that at the last glacial maximum, Antarctic sea ice was much more extensive than it is today. And we also know that kelp can't survive ice scour. So one hypothesis would be that sea ice knocked kelp off these subantarctic islands. And then as the Earth warmed up and the ice receded, the kelp was able to raft around in the Antarctic circumpolar current and recolonize those subantarctic islands. But the problem with this hypothesis is that sea ice wasn't thought to have extended as far as many of those subantarctic islands. So see, there's Marion there, Crozet, Gough Island and Macquarie Island. None of those islands were thought to have been affected by sea ice 20,000 years ago, but we find evidence of recolonization there. So first of all, I threw this hypothesis out the window and I thought, it can't be sea ice, it must be something else that explains what drove the kelp to extinction in the subantarctic. I played with the ideas of temperature and sea level changes, but nothing fits as well as the hypothesis of sea ice. So then I had a closer look at the data that was used to make these estimates of sea ice. This picture summarizes that data. Each dot, each coloured dot, represents a site where geologists have taken a core of the seafloor. And they look at the core and they date different sections of the core, and they find fossils of diatoms in the cores. And diatoms are single-celled marine organisms with a shell or frustule made of silica. So they fossilize quite well, and you can recognize different species thousands of years later. And some species of diatoms are more common in sea ice than in the rest of the ocean. So where scientists find lots of those fossil diatoms, they say that there was lots of sea ice above that area of ocean at that time. But what you can see from looking at this map is that the dots don't cover the whole of the Southern Ocean. There's all these areas where there just aren't any dots. The blue dots are where scientists have found lots of evidence of sea ice. The yellow dots are where scientists have found no evidence of sea ice. But there's big gaps between the blue and the yellow, like here. And here's a huge gap. So actually, we're quite unsure where the edge of the sea ice was at the last glacial maximum. So I published a paper suggesting that perhaps sea ice at the last glacial maximum was more extensive than scientists had previously believed, based on the results from the kelp study. And I think Jackie's going to talk to you a little bit more about publishing in Science.
Thank you, Crid. Okay, so you can imagine a 3-year journey. You're doing your PhD and you get to the end of it and you find that you have evidence that suggests that maybe the traditional view about things is not right. How do you go about publishing that? How do you go about getting someone to accept and publish an article that says, I think the traditional understanding here might not be right? So what we're going to have a look at is that process that Crid went through. And what you've been doing over the last couple of years is you've been learning how to actually reference other people's work. And you've been learning that when you're writing and you're taking evidence from a variety of places, that your teachers are saying to you, what's the validity of that evidence? How are you sure that that information that you're getting is information that really can be trusted? And one of the things that you've learned over the last couple of years is that you should be looking to peer-reviewed scientific evidence. You should be looking to journals where the scientist has had to go through a process of peer review before they can actually publish the work and say, we're sure of it, so that you as the person who is reading it at the other end can say, yes, I can be confident that this person's work is valid. How does that process work? Okay, well what happens is that as you've heard, Crid went through a whole process of collecting data and getting to the point where she thought she had something, a story to tell about the process that she'd been through and the findings. So she would then select a journal, and she'd want to select a journal that was clearly going to be of interest to this work, was going to be relevant to that journal. And so she would write her paper up and send it off to the journal. Journals have editors and they have a number of reviewers who are all scientists who are experts in their particular fields. So Crid's paper would then be sent off to a number of reviewers who would read the paper and make comment on it and send those comments back to the editor. The editor then has the option of what decision he or she will make. Now, it might be that they say, yes, look, everyone thinks that this is an excellent paper. We'll totally agree with the way you've written it, except let's publish that paper. Okay, how often do you send something into your teacher and you get it back immediately, a major assignment, saying, no comments, fantastic, absolutely brilliant? Not very often, right? Okay, well, things aren't going to change when you become a professional. Whatever profession you go into, you're going to find that that's an unusual situation. Normally, someone just thinking of it with a different set of eyes, different mind, might say, okay, you've got something interesting here, but we want you to think about X or Y. Hopefully that's what they do. Hopefully they're not going to go, no, look, you're really way off the mark. You have no evidence that can really support anything that we would accept here, reject the paper. Now, if by any chance you did get the paper rejected, but you were still convinced that you had something worth publishing, you're just going to go round and find another journal, because it could be that that journal said, look, this really isn't of interest to our readership. Okay, let's hope that the editors has come back to you and said the reviewers have got some comments for you, and you will get a copy of those comments, and the reviewer might say, look, I want you to give me more evidence of X, or, you know, I really am not convinced by the statistical analysis that you've used, or have you thought about another person's work and looked at whether there are some links here. So they're going to request some changes, and you're going to take that back. You're going to talk in your scientific group about the comments that have come back, get some advice from those around you, make those changes, and then it will go off to the journal again, and it will be sent again to the reviewers, who will look at it a second time and make some more comments. Now, hopefully it doesn't go round that circle too often. It could go round that circle 2 or 3 times, but hopefully you actually get to the point where the changes are made and you have a publication. And so that's what CRID has done, and that is what you're reading, the opposite end of that, when you're looking for evidence to write. For instance, if you're doing your 3.2 task and you're having to present evidence and you're having to describe to your teacher the validity of that evidence, that is why you really want to be looking to a peer-reviewed journal and saying, yes, look, the scientists have been through this process, they've published, therefore I can be reasonably confident that there is evidence here that can be believed. When you do your own investigations, that is the kind of process that you're going through in your own school with your teacher and the teachers that are working within your school, who will make comment and feed it back to you before you might go and take that to a local science fair or to the Royal Society's Realise the Dream and say, look, I'd like to actually put this out in public and publish in the way that you can as a school student. Now, CRID's work went a little step further. When a scientist actually grabs the attention of the mass media, we know that really they have hit on something that is impacting on the way that the general public understand science and see science. And that is exactly what happened to CRID, because what you can see here is a whole series of headlines. Kelp genetics reveals Ice Age climate change clues. There's one down the bottom there which is in French. I'm not going to try and read it. Or is it Spanish? No, it's French. It's French and it's wrong. Now that's the other really worrying thing for the scientists. The last ice age colder than we thought. And so what CRID had done here was not expected at all to look at the ice age and the extent of the ice in that period, but had actually, through her process of her PhD, which is not something that never happens, it's something that does happen from time to time in scientific research, as well as finding the answer to the question that she was asking, she also was producing some evidence which was useful in a broader sense. And in this situation, it was around the understanding of those climate cycles. So CRID is going to come back and tell you what conclusions she did make and did find from that work.
Thanks, Jackie. So yeah, that final headline there in French actually says that I said that sea ice went as far as New Zealand at the last ice age, which I certainly never said. So it also goes to show the media can get things completely wrong and you shouldn't believe everything you read in the paper. So I set out to ask this question: does De Villiers Antarctica disperse via rafting? And certainly this dominant haplotype throughout the subantarctic suggests that it's getting around. Those have all come from a common source population not too distantly in the kelp's history. So we do think that rafting has helped kelp to disperse. But the high levels of genetic diversity further north suggest that rafting is not the only thing that's driving genetic patterns in kelp. In fact, we think that kelp was wiped out in the subantarctic by sea ice at the last glacial maximum. If this is the case, though, sea ice must have been more extensive than previously believed. So this has shaken up ideas about what we know about past climate change. And why do we care what happened to kelp 20,000 years ago? Most people don't even care what happens to kelp today, except me. But understanding the effects of past climate change on plants and animals and other things will help us to predict the effects of future climate change. I guess the problem here is that predictions say that climate change, global warming, is going to happen much faster than previously in Earth's history. So how plants and animals are going to be able to respond to these really rapid changes will probably be quite different to how they've responded in the past.
Thank you very much, CRID. Now, what we're going to do now is go to a question answer time. Just before we do that, we're going to remind you of the challenge questions that are there for you. Now, we have 3 sets of challenge questions for you this time. The first set is looking at the concept of species. can we emphasize that the data that Crid is presenting to you here is all around the one species, Davilia antarctica. There is variation within that population which she has found. So what we want you to do in this first series of challenge questions is really identify what biologists mean when they talk about a species, how they actually decide that we have a differentiation in a genus, and we can actually say, yes, we have two different species here. It is not just variation within the population. And we want you to explore a little the differing ways in which we can use different evidence to actually define species. So that's the first set of challenge questions, and it relates to this diagram here, which is, of course, Crid's data which she has presented to you about the one species, Davilia antarctica, and the variation that she found in the populations, and the way that she found that that variation could be seen to be geographically distributed into a pattern. Okay, the second challenge is around terms and techniques. So in the second challenge, we're going to want you to look at the term infer. It is not unusual for scientists to actually look at the idea of what does the term infer mean, to say that we're inferring something. So what we want you to do is look at what the term infer means and why in this particular context they have used the term infer. Okay, I think we can hear welcome Otago. Okay, so we'll just finish going through those. And hello, Otago, that is Dunedin that we can see down or hear at least. Okay, the third set of challenge questions is the one where we're going to actually ask you to analyse some data. And the first thing that we're going to ask you to do is to explain why the evidence suggests that the population variation that we see, that population dominating the sub-Antarctica at that point, is actually a very recent colonisation in geographic time. So that's looking at the circular diagram that is taking us through the way in which that population was spread out. And then the final challenge is to actually look at this new data which Crid has put up for you and to analyse what you think is happening with the different parts, the variance within that population, those populations, and see if you can explain to us what is happening there. So those are the challenges, and we're really looking forward. Michelle and Crid and Hamish are looking forward, as I am, to hearing your responses to those and seeing how you get on with answering those challenge questions. So now we're going to go to a question and answer session. I'd like to invite Crid and Hamish to come forward, and we are going to look at a variety of questions. We've got Otago. Welcome to Otago down there. Can we cross to Otago and they can actually say hello? They're there? Right, okay. Otago, which schools have we got represented down there?
Logan Park High School.
Logan Park High School, welcome. And anyone else?
What else have we got?
All from Logan
Otago
Park
girls.
High School. Well, Logan Park High School, that is absolutely fantastic. Otago girls, are you not there?
Anyone else?
Oh, you are there. Oh, okay, you're at the back there. I was sure Otago Girls was also joining in. So if you're from another Dunedin school, remember that there are a group that meet at the University of Otago, and it's fantastic that we have Otago here with us live in a day when we have scientists here from the University of Otago. So we're looking forward to your questions. Maybe Logan Park and Otago Girls, have you got a question for us to kick off the question answer session?
Yes, somewhat. Kieran's left now, but he was wondering if different parts of DNA were more susceptible to mutations. So with the phylograms and working that sort of thing out, do different bits of DNA sort of change more quickly and that sort of thing?
Okay, that's a very good question. So when we're looking at the phylogram, we're looking at variation within the DNA. And so the question is, are there different parts of DNA that are more susceptible and will change more easily than others? Okay, so handing over to you, Crid.
Okay, well, yes, there are parts of DNA that are more susceptible to mutation than others. Often it's to do with which parts of the DNA are used. You know, a lot of your DNA is not actually performing a lot in your body. It's just junk sitting there. So the bits that are producing proteins are going to be much more important and much more likely to be conserved than the other bits. But what's interesting in the variation I found in kelp, I found 117 variable sites across a 600 base pair region, and pretty much all of them were synonymous changes, so they didn't actually cause any change in protein that would be produced by that part of DNA. So you can have variation that doesn't affect the DNA, and that's often what we're looking for when we're doing phylogenetic analyses. Is that enough?
Okay, thank you, Chris. So what you're talking about is when you have variation that doesn't actually give a change to the protein that is produced. And that is what you can have. Don't you love it when scientists talk about junk DNA? I think it might mean DNA that we're not sure what it does at the moment. Excellent question. We'll come back to you in a minute, Otago. We're going to go to a question from Tom Norton at Nelson College, which came in before the seminar. Tom asks, how do scientists choose target genes to analyse when investigating the phylogeny of related organisms? And it's a very good question. How on earth do you decide? Where is my target going to be? So, Crit, if you could take us through the process there.
So I went through this a little bit in the talk, actually. A lot of it's chance. A lot of it depends what you can get to amplify and which bits of the DNA turn out to be informative. But you do set out with some ideas in mind, like lots of previous studies have used a certain gene region, so you might try to target that if it's shown to be informative in other taxa. It also helps if they've been used in other taxa because there are probably primers available that you can at least start to get something from the DNA from, and then design better primers after you've seen that. And then, of course, choosing whether you use mitochondrial or nuclear, or in plants, chloroplast. Those sorts of DNA are inherited differently. As you know, mitochondrial is inherited maternally, and nuclear DNA you're going to get crossing over. So it's important to think about those things when you're deciding what questions you're trying to answer.
Thank you very much, Crit. And of course, one of the challenge questions we have for you is to actually think about that concept of, would you use nuclear DNA or mitochondrial DNA, and what would be the pros and cons of each? Now, I'm going to cross now to Michelle, who is down on the wiki chat or the live chat today. Michelle, have you got some questions coming through for us that you'd like to put through to us now?
Hi Jackie, yes we do. They're firing through very quickly. My first question is from Cherry at Hillcrest High School in Hamilton. This one's for Hamish. Can you please explain in a little bit more detail the difference between the top and bottom numbers on each branch of a phylogenetic tree?
Okay, I feel like I'm a bit on a quiz show, you know, and this question has come in and I don't know what it's going to be about. But those two numbers, the one at the top was the one that went between 0 and 1, and I called that a posterior probability, or sometimes a Bayesian posterior probability. And the way that that number is calculated is that the program goes and finds a tree, and it records all the branches that are in that tree. And then the program goes and changes that tree a little bit and asks, is that a good tree given the data that I've got? In other words, is that the kind of tree that would generate the data that I've got? And if the answer to that question is yes, then it says, well, that's a good tree. Let's see which branches are in that tree. And if the answer is no, it throws that tree away and goes back to the original one and does another little, what's called a branch swap, where it takes a bit of the tree and changes it a bit in some way. And there's different ways of doing that. And again, it asks, is that a good tree? And if the answer is yes, it keeps that tree and it asks which of the branches are in that tree. And at the end of doing that lots and lots and lots of times, it looks to see what proportion of the branches in the tree that it thinks are the best are found in that huge collection of trees that it got by rearranging, right? So clearly, if all of the good trees, the trees that it kept, have a particular branch, then there's good support in the data that you have for that particular branch. And it just scales that number between 0 and 1. So it's the proportion of times that the algorithm has trees that have those particular branches in them. Right? So that's how the Bayesian probabilities work. The likelihood values use a completely different idea, the bootstrap values that were underneath the branches. Remember, they went between 0 and 100. And bootstrapping is a very, very clever idea that was invented last century, where you go and you resample the data. So it says, this is the DNA sequence that we have. Let's go in and sample from that DNA sequence. Let's pretend that we are actually going out and taking a sample of individuals from the data that we already have. And let's take that sample and construct an evolutionary tree from that sample. And let's again go and look and see which of the branches in the tree that we think's the best are found in that tree that we've just had from the sample of the data. And let's go and do that again. Let's go and take another sample from the data we already have, construct a tree from that sample, and look to see which of the branches are in that tree from the subsample. So these samples are called pseudo-samples, right? So you take a lot of these pseudo-samples and you construct for each pseudo-sample a tree. And you look in all of those collection of trees to see which of the branches that are in the tree you think are the best are in that sample of trees based on all the pseudo-samples. So again, you can see that if that number is a large number, if it's found in all of the trees constructed from the pseudo-samples, you think that the data really support the branch that's in that tree. So there's a completely different ways of coming up with an idea of how much support there is for the particular branches in the tree. But they use a kind of similar idea. They look at a bunch of different trees and they ask what proportion of those trees contain the branch that you're interested in.
Thanks, Hamish. And of course, what we've got there is the power of mathematics applied in a biological situation to actually help us analyse the data, and thankfully, the incredible power of computing that allows us to deal with such massive amounts of information. Okay, now I'm going to go to the live audience now and ask them whether there is anyone that has a question. And then from the live audience here in Auckland, I'm going directly back to the live audience in Dunedin. So if you've got a question and can be ready there, Dunedin, and then we'll go back to the Wiki chat. So have we got any questions from the live audience here in Auckland? Would you like to go to the microphone? That's fantastic. Would you like to tell us who you are and where you're from?
I'm Emily from Auckland Girls Grammar School, and this is a question from our school. Can the rate of mutation change, like speed up or slow down the, and what would cause a change to the molecular clock?
Okay, so can the rate of mutation change, and can the molecular clock itself actually change? Okay, so Hamish probably...
Well, the short answer to that question is yes. The rate of mutation can change, and you might have noticed on the phylogram that I had there that all of the branches that were of different lengths, all the species that were there in the present time didn't have the same total length of branch to them. And that's because there's a little bit of variation along different branches of a tree in just how fast the molecular clock is ticking. So there can be changing. The original molecular clock assumed that the rate of change was constant all the time in all the branches of the tree and so on. But now with modern mathematical techniques, there are molecular clock techniques that don't make that assumption. So you can adjust for different changes along the different branches, different differences in the mutation rate.
Okay, what a very good question. Thank you very much. So we're going to cross now back down to Dunedin and see whether we have another question from our audience in Dunedin. Have we got someone down there that has another question for us?
Yeah.
Yep, okay. Excellent. Let's go with your question.
How much variation does there have to be in the DNA for something to be classed as another species?
Okay, very good question. And is that Otago Girls or is that Logan Park there?
Otago Girls.
Otago Girls. Okay, thank you, Otago Girls. So the question is, how much variation does there have to be within the population in the DNA before you're actually starting to look and say, have we got another species here?
Okay, that's a very good question and also a very difficult one to answer. And I don't think there's any one answer. There's not a number that defines how different clades or lineages would have to be for you to say that they're different species. I think you need to look at a whole lot of other things as well. Often you look at the morphology, so how the plant or the animal looks. It might be a different colour. Sometimes things are different colours, but they all get grouped together originally because they can't find any grounds to break them off. And then you might find when you look at the DNA that actually all the black ones are very genetically different to all the grey ones. So then there's good evidence that they are separate. I think in the kelp that I was looking at, I got differences of around 4 to 5% for mitochondrial DNA between some of those lineages in De Villiers, Antarctica, and that would normally be enough for us to start thinking, yeah, these are probably separate species. Another good test is whether they can interbreed and produce viable offspring, but with something like kelp, that's quite tricky because you don't always have the years that it takes to breed kelp in the lab. You know, they get quite big, and then you've got to see if that one can also produce sperm and eggs. That can, so in the end, we sort of do use a lot of genetic, and we look at the geographic regions where they occur, and so we can make some decisions based on that. But it is a hard one to draw the line. Would you add anything to that, Hamish? Or?
No, that's
No.
a good answer. Hard question and a good answer.
Yeah, thank you very much, Otago. It's a very good question to ask, and some of the best questions are the hardest ones to answer. And in fact, one of the challenge questions we've put to you is to actually start thinking about and looking at what is the evidence that we start to use. What are the different types of evidence that we can use when we're wanting to identify whether we have, in fact, created two species. Okay, we're going to go to another question that came through from Jenny at Unlimited down in Christchurch. This species of kelp is unique because it floats by a honeycomb-like structure, which is unique. It prevents the kelp from being damaged by strong waves. Is this further evidence that the kelp is more likely to be dispersed by currents because it survives for a longer time? Corinne, would you like to address that one?
I think certainly the toughness and the buoyancy of the kelp is helping it to survive for a long time, which will help it to get around the ocean. Another good thing about seaweed, another reason why it's probably able to disperse, is that unlike plants, seaweed doesn't have roots. It doesn't need to be attached to the earth to grow. So it can absorb nutrients from the water, and of course it uses sunlight to get its energy. So a kelp drifting at sea should really be just as likely to survive as one attached to the rocks, although there are fewer nutrients out in the open ocean. Whether other species of seaweed would be as good at dispersing, I think ones that are less buoyant and also less strong would be more likely to, well, sink obviously, and also get torn apart by the waves. But Davilia antarctica is a big kelp. It's almost part of the habitat that you get along the intertidal. So you can often have other seaweed species growing on the kelp. And then if Davilia antarctica can raft across an ocean, some of these other seaweed species and other animals might be able to travel with it.
Thank you very much, Corinne. And again, thank you, Jenny. A very good question to get us thinking about those issues. Michelle, can I go to you and can we have some more questions coming through from the rest of the country, please?
Hi Jackie, I've got a question from Amber down in Wellington Girls College, and this is for Crid. Why did you choose to study bull kelp for your PhD? What made it so interesting?
Oh, of course, I've always loved kelp. No, I haven't. I never really thought about seaweed until I came to New Zealand, but I was looking for a PhD. I'd finished my undergrad. I was doing my honours in Australia at the Australian Museum. And at the time I was working on worms, which are just as interesting as seaweed. But I was looking for several things when I was looking for a PhD. I was looking for a good supervisor or good supervisors, people I could talk to easily and who would be there to answer my questions, because I did speak to some people, students who were working with supervisors I was interested in, and they said, oh, don't work with them, they've never got time to answer your questions. So you want to look at different things when you're looking for a PhD, and a really important one is your supervisor, whether you can get along with them and whether they'll be there to help you. I was also really keen to get to Antarctica or the subantarctic islands. So that was something I was looking for when I was looking for a project that would take me to some of these places. And I did work, before I started my PhD, I worked on penguins. I love penguins. But biologists do some terrible things to the organisms they work on. So we used to have to flush the stomachs of the penguins and things like that. So I found it was a bit distressing working on something you loved so much, and I decided to move to something that would let me see the penguins without actually grabbing them and squeezing their stomachs and distressing them. So that's how I ended up working on seaweed. I was sort of Googling and trying to find people who were working in fields I was interested in. Another factor, I know it sounds strange, but I was keen to get out of Australia and out of Sydney. It's too big and noisy, so I was looking for somewhere. Dunedin's perfect. It's nice and quiet and relaxed. So all those sorts of things come into play when you're looking for a project. And just by chance, this project was, and actually when I started, I wasn't planning to work on the seaweed. I was working on the animals that live in the holdfasts of the seaweed. But bit by bit, as I worked on the project, the seaweed grabbed my attention. I started to get some really exciting results. I was initially just playing with it as an extra in the lab. But the results coming in were so exciting that it consumed my PhD, and in the end all my chapters were on the bull kelp itself.
Thank you, Crit. And it is. It's a journey, and it's something that you've got to think about. It's the beginning of a significant part of your career. You're going to be living and breathing the team and the group that you're with, and so it is really important that not only the subject interests you, the concepts interest you, but also you're with a team of people who you think you're going to learn a lot from. Okay, Michelle, we've got another question here from Jenny at Unlimited, and then we're going to go back to you. So Jenny asks about the different temperatures in currents. Some currents are warm or cool and rise and fall. They also reverse in El Nino, El Nina sort of events. Would these variables have a significant effect on the dispersal patterns of the kelp? So that's quite a complex question. It's an interesting question, Jenny, and I'm going to put it to Crid, who's been having a think about this one for you.
Okay, well, the question of different temperature of currents, whether they're warm or cool, that's certainly going to have an impact on where the kelp, not necessarily where it can disperse to, but how well it can survive. There are studies that show that kelp loses its reproductivity. It becomes less reproductively viable at warmer temperatures. Especially Davilia antarctica, it thrives in the really cold areas around the subantarctic, around New Zealand. You don't find it up in the tropics. So if some current, like the Humboldt current along Chile, takes it up towards the tropics, it probably won't survive, won't be able to colonize that region. In terms of El Nino and La Nina events, those tend to happen over a period of several years. So it could help the kelp occasionally reach places it might not otherwise reach. But in the long term, what we're looking at with these phylogeographic studies are patterns, processes that drive patterns of genetic variation over thousands of years. So they're unlikely to have an effect on the bigger scale, I guess.
Thank you very much, Crid. Okay, Michelle, I'm going back to you because we've got a lot of people out there, I know, and so if you can give us another question from somewhere around the country via the chat.
We certainly can, Jackie. In fact, we've got so many, we're trying to combine a few together. The first question here comes from Theodore. Crid, did you find any kelp in Antarctica? Because there were none shown on the diagram. And related to that, this was a combined one with Gemma from Waiuku College. Does the habitat in which the kelp is typically found have any effect on whether the kelp is buoyant or not?
Okay, so two ideas there. One about whether the Davilia antarctica or the kelp in general is in Antarctica, and the other around the habitat and its link to buoyancy.
Okay, I didn't find any kelp in Antarctica. It doesn't, this particular species of bull kelp doesn't occur in Antarctica. Probably because of sea ice scour. It does occur on the islands where sea ice occasionally reaches, but not always. So, for example, South Georgia has a few glaciers on it, and where those glaciers break off and chunks of ice float into the ocean, bull kelp does not grow. But there are pockets of that island which are protected from ice scour, and in those little pockets you get Davilia antarctica growing quite happily. So it seems to be driven by, its limit is dictated by the limit of ice. And obviously Antarctica is strongly affected by ice, so it can't survive there. In terms of whether the habitat of the kelp could affect its buoyancy, I think it probably can. There's not a lot of research that has looked into this yet, but there have been several solid-bladed or non-buoyant species described from places, subantarctic islands, southern Chile and the Patagonian fjords. And then when we've looked at those, they've genetically turned out to be identical to other buoyant Davilia antarctica. And I think maybe what's driving that is around these subantarctic islands, the water is much clearer than around New Zealand, for example, or along the coast of Chile. And maybe when the water is that clear, the kelp can occupy a deeper habitat. And so non-buoyant individuals are able to flourish. But normally they need to be buoyant to keep them up the top so they're near the sunlight. In New Zealand, if Davilia antarctica was not buoyant, it would probably not get enough sunlight. It would be down in the murky water.
Okay, thank you very much, and some excellent questions coming through there. Now, I'm aware that we have quite a lot of questions that we have not had time to answer today. We've had a fantastic 20 minutes or so of questions and answers, but we've still got more coming through. Now, we can't keep going, but what we will do is that anyone who has put a question through via the live chat session, anyone who has put a question through to the wiki in advance that hasn't been answered, anyone down in Otago who has some more questions for us, please feel free to put those up on the wiki as questions. Anyone here in the live audience with more questions, please do also put those up so that we can all see them. And we'll do our best to get some answers to those questions up to you as soon as possible over the next few days. I have really enjoyed being joined today by Crid and by Hamish. Thank you very much for your time with us. I think you'll agree that Crid's journey has been a very interesting one, and that you can see how the concepts of evolution that you're exploring at school are ones that she has applied and used to help understand what is going on here. I hope that it's helped you see how a scientist goes about the process of the early parts of their training, and the process of communicating that and getting some validity in peer review about the findings that they have and the confidence that we can have when work is published. Now, we have a good number of challenges up there for you. Michelle will be leading you through that challenge period over the next 3 weeks. We're really looking forward to your answers. We're looking forward to the discussions that you have within the wiki, within the challenge questions, and we'll be coming back to you after the term break with a seminar that will be looking next time at biological clocks and some very interesting work that is coming from the Faculty of Medical and Health Sciences here at the University of Auckland. So thank you very much to the Alan Wilson Centre for hosting with us today, and to everybody that has participated. Good evening. Everybody, I think we're